Welcome guys, today we're going to go over transcription and translation, again in preparation for the Bio EOC. Specifically, our objective is to understand the processes of transcription and translation and to be able to compare and contrast the two. So to get us started, let's do a little review question. Simple question. True or false? DNA consists of a phosphate, sugar, and a nitrogenous base. Take a second to answer that. The answer is true. That's true. DNA does consist of those three components. So here I have DNA and I have RNA. Well, let's look at DNA real fast. It's got three parts to it, three basic parts to it. It's got a phosphate, which is designated over here, a sugar. It's got one of four bases. In this case, I've got T, which is thymine, and thymine always pairs with A or adenine. Also, I've got a C or cytosine, which pairs with G and guanine. So DNA is double-stranded, has these three basic components that make up something called a nucleotide. So all three things together, the phosphate, the sugar, and the base, are known as a nucleotide. RNA is a little bit different. What's striking about RNA initially, from initially looking at it? Well, RNA has only one strand. DNA's got two. So RNA is single-stranded. Also notice that RNA doesn't have T. Instead, in its place, it has U, or uracil. So RNA's got U, DNA's got T, or thymine, and that's an important distinguishing feature of RNA and allows you to identify immediately that you have RNA and not DNA. And you will see when that comes in handy as we go more in depth over these two processes of transcription and translation. So over here I have the life cycle of DNA. DNA can really do one of three things. It can replicate, go through transcription, and ultimately translation. Let's talk about all these three things individually. So replication is basically where you make more of DNA. Now, why would you make more of DNA? Why would you want to make more of DNA? Well, really, when you create more cells, because remember, DNA is in the nucleus. So that leads to my next question then: when do you create new cells? Remember, you create new cells for periods of growth. Could be actual physical growth, growing taller, or it could be growth of hair, skin, okay, fingernails. All that is mitosis. All that requires new production of DNA within a newly made nucleus. Okay, so replication can be for that reason. So what's another reason you may have replication? Well, for healing. Say you have an injury of some sort, a cut, a broken bone. In these cases, you'll need to make a lot more cells, and they're going to go through replication. Also, you can take that DNA, you can create RNA. The creation of RNA from DNA is called transcription. Okay. Finally, taking that RNA and making protein, which is the whole purpose of having these coded instructions with this DNA, okay, is going to be to make that protein. It's going to be through translation. So production of protein is the purpose. You are the results of your DNA. Okay? I'm not saying every characteristic, but the core of who you are results from your DNA. Who do you get your DNA from? You get it from your parents. So your parents give you your DNA. They pass it down to you. You get half from your mom, half from your dad. And that DNA becomes protein, and that protein then will influence traits, physical as well as personality traits. Okay? So that's kind of a broad overview of what we want to go through today. I'm not going to go through replication. That was done in a prior lecture when we talked about DNA. But I am going to go through transcription, and I am going to go through translation. So in transcription, as we just said, I'm taking DNA, I'm going to use it as a template, I'm going to make RNA. Okay, specifically, I'm going to make messenger RNA. So here's a very rough diagram, but it gives you a good a good idea of what's happening. So here is my double-stranded DNA in the blue and the purple. I go through it and I create mRNA using RNA polymerase. Okay, so RNA polymerase is an enzyme that uses DNA as a template to create mRNA. And how do I know I have mRNA? Well, the smallest individual units, or the nucleotides, are going to have, again, a phosphate, a sugar, and then one of four bases. A, U, C, and then G. Notice RNA has U, not T. That's the distinguishing feature of between RNA and between DNA. So here I have now transcription. Again, same exact process except a better diagram. Here I've got my DNA. Okay, in the background I have my DNA. I've got two strands. After uncoiling one segment, I'm going to use one strand as a template to make my messenger RNA. How do I know this is messenger RNA? How do I know it's RNA in general? Well, I know that because instead of T, I've got U. And the U base pairs with the A. So this is DNA. How do I know this is DNA? It's got T. It's got the thymine. So then I go and base pair them, though. And again, it's not shown, but RNA polymerase is, in a sense, the matchmaker during transcription. It matches the bases together. A with U, T with A, G with C, C with G, and then T with A. Now, these are laid down as nucleotides, meaning although it's not shown in the diagram, that U, that A is also going to be connected to. Each of these will also be connected to a phosphate and a sugar. Okay? They're ribonucleotides because that sugar is ribose. These would not be ribonucleotides up top. They would be deoxyribonucleotides because DNA has a sugar, deoxyribose. So that's transcription. That's going from DNA to RNA or messenger RNA. Now, the messenger RNA itself then has to be made into the protein. And what was the monomer of a protein? That's right, the monomer of a protein is a amino acid. So in actuality, you're taking that RNA and you are translating it into an amino acid. So the genetic code, though, is read 3 base pairs at a time, or 3 letters at a time. So the RNA will code for an amino acid, but it will take 3 base pairs, or 3 codons, 3 base pairs, or 1 codon, to code for an amino acid. So 3 base pairs will code for 1 amino acid. The name for those 3 bases is a codon. And let me simplify that. Let me show you charts. Here I have a chart of base pairs. Immediately, you should be able to tell me, is this RNA or DNA? That's right. This is going to be RNA. Okay? So this is RNA. How do I know that? Because there's U, and do you see any T within these triplets? No, you don't. So you know you have RNA. And RNA, again, will code for amino acids. How many base pairs of RNA? 3. 3 will code for an amino acid, 3 for amino acid, 3 for an amino acid, 3 for an amino acid. Same with these. I've got these two codons will code for the same amino acid. So that brings up a good point. I've got 2 codons that can code for PHE or phenylalanine. I've got 4 codons over here that can code for serine. In actuality, there's 2 more over here, so a total of 6 codons that code for serine. So the genetic code actually is known as degenerative. What does degenerative mean? It means repetitive, meaning there are multiple codons that can code for a single amino acid. So let's say there was a mistake during the process of transcription. There's a mistake, and instead of UCU, I laid down, or your body, I should say, laid down UCG. Is the protein going to be misshaped? The answer is no, it's not. because UCU and UCG are both going to code for serine. Okay? So built into that code, there is the possibility to make some mistakes and still get the amino acid that we want. Now, notice these amino acid sequences. They don't code for anything. Instead, they code for something called a stop. This one does, these two do, as well as the UGA. They code for stop codons. So what do you think a stop codon is? Well, let me tell you. Stop codon is going to stop the process of translation, or the process of protein production. So at some point, your body is going to get to a certain amount of amino acids, and then it's going to get to one of these. Once it hits one of these, it will not be able to translate it into an amino acid. Instead, it's going to stop. So these codons will stop the process. So if there's a stop to this whole point, there has to be a start. The start codon is known as AUG. What does AUG stand for? It's going to be methionine. So AUG will be for methionine. Every single protein in your body starts with AUG or methionine. Why? Because this is the code that starts translation. You have to have this code to even start the process of translation. You always start with methionine. Okay? So let's try this question out. I'm going to read the question. I suggest you stop the video and you try it out on your own, and we'll go over it together. Okay? So I'm going to go ahead and read the question for you. What is the function of the coded instructions contained in the body cells of an organism? Okay, so go ahead and answer that. Alright, so let me keep going with this. So what is the function of the coded instructions contained in the body cells of an organism? A. They direct the synthesis of proteins necessary for proper cell function. B. They synthesize different kinds of amino acids in a specific sequence. C. They form a variety of gametes that will pass on hereditary information. D. They produce inorganic molecules needed for normal cell growth. Okay, so I kind of flew through those answer choices. Let's go through these one by one. And I'm going to start with D. So they produce the inorganic molecules needed for normal cell growth. So let's read this question again. What is the function of the coded instructions contained in the body cells of an organism? Coded instructions refers to what? Well, you should know immediately. The code, genetic code, is DNA. What does DNA become? Protein. So DNA ultimately will be made into protein. So knowing that, let's see what we can do here. So they produce inorganic molecules needed to for normal cell growth. So in a prior lecture, we talked about macromolecules, and one of those macromolecules is proteins. What are all the macromolecules made up of? Well, they're made up of carbon, which makes them all organic. They're carbon-based and organic. So D cannot be the right answer, because the proteins that are made ultimately from DNA are organic and not inorganic. Let's look at C. They form a variety of gametes that will pass on hereditary information. Does transcription or translation have anything to do with the production of gametes? Well, gametes are eggs and sperm, so no, nothing to do with that at this point. What produces eggs and sperm? Meiosis. Okay, and that's a completely different topic. So C and D are excluded. Let's look at B. They synthesize different kinds of amino acids in a specific sequence. That's not a bad answer. So we'll come back to that. Let's look at A. They direct the synthesis of proteins necessary for proper cell function. Also, not a bad answer. So knowing what DNA is, what does DNA do? What's the point of DNA? Is DNA itself going to synthesize different kinds of amino acids? Does the DNA create or make different kinds of amino acids? Or does it direct the synthesis of proteins necessary for proper cell function? Well, DNA itself is going to be more of a code. Codes are there to direct. What actually makes the different kinds of amino acids? we're going to see that it's a ribosome. Okay, the ribosome is what's actually going to go ahead and create a sequence of amino acids, or the protein. The code will direct or be the foundation of that synthesis of proteins. So let's try another question out. This one might be a little bit easier. So codon is a sequence of 3 nucleotides that code for an amino acid. If the DNA sequence shown here goes through transcription and translation, how many amino acids will result from this sequence? So I've got different nucleotides, and remember, what's not being shown is a phosphate bonded to a sugar. I know this is DNA. Why is this DNA? Because I have thymine. Now, if each amino acid is a triplet, how many amino acids are there? Well, I've got 1 triplet, 2, and 3. There will be 3 total amino acids, and the best answer is D. Let's say there were 6 triplets. How many amino acids would there be? Well, there would be 6. How many base pairs would there be? 18. Okay, so that's a fairly simple question. It's a question they love to ask. Okay, because I don't know, it's people get tricked up. Biology is not too mathematically orientated, so once people throw in a math question, people get tricked up. So we talked about the stop versus stop codons. How many stop codons are there? 3 stop codons. How many start codons are there? 1, and it is methionine. Don't forget that. Do not forget that. So translation, though, is when you take that ribosome and it's going to match an amino acid to the 3-letter mRNA codon. So in blue is my ribosome. What's the function of the ribosome? Well, in this case, the ribosome is actually going to match the mRNA to the amino acid. And it does that with the help of something called tRNA, which is known as transfer RNA. So as the word implies, the transfer RNA is basically going to transfer the amino acid to its specific spot. So is this going to be the end of, what do you think this is? The end of translation, the middle, or the beginning? Well, to be honest with you, it's most likely the beginning, because what's my first amino acid? Always. It's going to be MET bonded to the codon AUG. So here's my first amino acid brought in by the tRNA. Now the next one can also get into the ribosome. And in this diagram there's only 2 slots. Some diagrams have 3 slots. But that tRNA will be directed into this slot. And how's it going to know where to go? And how's it going to know which specific amino acid to bring? Well, it's going to read the codon. GCC, let's prove that GCC goes to ALA. Well, where's GCC? I know this diagram's cut off, and I'm very sorry for that. But, try to visualize. Here's my G over here. There's my C, and there's my GCC. And that's going to go to alanine. So this diagram is accurate. So the GCC then will be the alanine, and this is bringing in, the tRNA is bringing in the alanine to bind to that slot, and then it allows connection of these 2. Next, I have my next tRNA coming in. It's going to be the TRP. Well, the tRNA can't get in, though. So what must happen? I've got 2 of them in 2 of these slots. This tRNA, how's it going to get in? It can't right now, so what's going to happen? One of these is going to have to be booted out. And it's going to be this one. This one on the left will get booted out, and this entire tRNA, which will then be bonded to the ALA and the MET, will move over to the left. Okay? As it moves over to the left, it will free up that open slot. So now I have my ALA on the left, the MET on that same tRNA, allowing an opening now for the next one. So basically the ribosome will shift 1 codon in the 3' direction. Now I'm not going to focus on 5' and 3' right now. I'll do that for another lecture. This is more of an outer surface kind of a biology EOC take. I will in my next lecture go a little bit more in depth and go over what 5' to 3' directionality is. Right now I just want you to understand what's going on in this diagram and be able to identify the components. Okay? So that really concludes this lecture. I hope it was helpful. Definitely go through this over and over again. You should be able to visualize this yourself. You should know some of the details, the finer details we went over. And if you do all that, you will be very successful on this part of your test. Okay? Shoot me some comments, okay, and any questions that you may have. Good luck studying.